Do I do a share screen?
This is your second.
Yeah.
I'm gonna use. You want to get rid of that on the end?
Yeah, absolutely. Everyone just look at me. Not the other people. I think we should be good. All right, thank you for the introduction. Yeah, so my start here has been a little on and off, but we've been back and up and running. I think I have been. I've been spending money since the fall of 2021. And that's so far so good. Still have some somehow. So today I'm going to talk about really the seed finding from my postdoc that founded my whole research program here at U of A. As Declan alluded to, it's quite a departure. I haven't always got to meet everybody yet because I'm still relatively new. So I did kind of keep in a little bit of background. As Declan was talking about, my master's was on neuroplasticity in the songbird brain. This is where I first fell in love with neurogenesis, which is a weird thing to fall in love with. I got way out there on my PhD and I studied nest building in birds, both the behavior and the brain behavior connection across evolution and in the lab within zebra finches. Then I made the forbidden switch from finch to fish. And in a huge departure, I went to U of T for a postdoc where I started studying sensory experience dependent brain development and could return to my love of neurogenesis, now with a much more convenient model. So most broadly, what my research program is interested in is depicted here. So these are MRIs of the same human being taken through the brain from 1 week to 10 years of age. I'm interested in development of the nervous system. Now, unfortunately for scientists, fortunate for this person, the brain doesn't develop in the black void of an MRI machine. It develops in a beautiful multisensory and complex world. And one way, and this is really the interaction I'm interested in, is how we kind of ecologically place brain development in the greater, more complex world. And one way I look through that is the interaction through sensory experience. So the brain develops in the world and can sense it and sample it and interact and co-evolve with it through sensory input. So how does sensory input impact developmental processes? So a brief outline, what I'm going to talk about today. I apologize if you saw my interview talk. This is going to be 90% the same, but I did try to put some preliminary data in there. So first I'm going to talk about a specific period of development that I typically focus on called post-embryonic development. That word, those words appear a lot in the literature, but we very much mean them in a very specific context. And so I'll explain that context and why it's important for our research. And then I'm going to focus on the biggest project that came out of my postdoc that motivated my will to continue in academia, which is looking at sensory motor dependent neurogenesis and development. So despite you all thinking lectures ended yesterday, you're going to get a real brief lecture to understand some specific form of zebrafish neurogenesis. I'm not kidding, it is very much like a lecture. Then I'm going to talk about the actual exciting experiments we use to connect movement and the sensory experiences evoked during movement with developmental neurogenesis, specifically in the forebrain of zebrafish, and how we actually broke down movement itself into different forms of sensory experience to identify ones that are actually responsible or mediate the effects of movement on brain development. And we're going to have a little preview of our slow creep to life here. All my grad students just started last fall, so this is the end of our second semester. But already, I'm incredibly proud of them, and they've already produced some very interesting paradigms, if nothing else. So post-embryonic development, like I said, can be used very generically. It really means anything past embryonic development. But we mean it in a very specific way. And I find the best way to explain it is by talking about both development of sensory systems, but also neuroplasticity. And the best way to approach that is to compare it with what comes before and after. So embryonic development and adulthood. So when it comes to sensory systems, embryonic development is characterized by the generation of a sensory system, but typically not the function of them, at least not in the nervous sense, the nervous system sensory systems. Contrast that to adulthood, where you have mature functional sensory systems that aren't, they're probably the least plastic that they will be in life. Post-embryonic development sits awkwardly between the two and uniquely captures a period where sensory systems and neural circuitry come online, quote unquote. So now you have circuits that for the first time in life are actually sufficiently developed to actually process incoming sensory input from the environment or from the body in this case. The second trait is neuroplasticity. Now, unsurprisingly, during embryonic development, you go from one cell to more than one cell, a lot more than one cell. So you have the highest levels of neuroplasticity in life. In adulthood, again, contrasting to that, the lowest levels of neuroplasticity and what we kind of see as the greatest stability that the nervous system usually can achieve. And the post-embryonic development, again, a weird intermediate. Elevated, albeit tapering levels of neuroplasticity. And it's the combination of these two traits that we care about the most. That you have circuits that for the first time have come online and start functioning, but at the same time are continuing to grow. Now, the evidence that this is a largely or kind of most predominant in post-embryonic development is the concept of the sensitive and or critical period. And these overwhelmingly occur during post-embryonic development, and they represent developmental windows in which a specific sensory experience or input exert their greatest impact on brain structure or function. So they have their biggest impact on brain development, and they overwhelmingly occur post-embryonically. Not surprisingly, because at the very least, you have to have the circuitry to actually process sensory input. The classic example, throwing back to my old days, is Peter Marler's really famous work on sensitive periods in song learning in birds. So in some species of sparrows, you can actually classify the life into these different kind of generic periods. And the first one is, the first post-embryonic period is the sensory period. What Peter Marler found is that birds that are tutored or hear an adult mature crystallized song during the sensory period will eventually go on to babble and learn and produce that song. So they're uniquely kind of tutored during the sensory period. If you present the same song outside of that period, even though the bird's not even singing yet, it won't actually learn it particularly strongly. There's a real sensitivity unique to the sensitive period, even though that period will close, and there will still be a couple months before the bird even babbles or tries to sing. It will try to sing what it was tutored in the sensory period. So what happens in the brain, and this has been studied for decades, what happens in the brain when you have one of these sensory experiences during a sensitive or critical period? Well, overwhelmingly and traditionally, we consider the changes to occur in pre-existing neurons. So pre-existing brain cells, just a couple examples of them from an Eric Knudsen review in 2004. You can get things like axon elaboration. So the projection of brain cells will elaborate. In some situations, they'll prune, they'll get smaller. Within each one of these kind of projections, at the end, you might actually see the addition or elimination of synapses or connections with other neurons and other brain cells. And even within each one of those boutons or projections or synapses, you can actually get changes in the form of things like strengthening and weakening through mechanisms like long-term potentiation and long-term depression. My issue with this approach is that it largely rests on an assumption that ignores the concurrent form of neuroplastic development, and that's neurogenesis. Now, traditionally, with the mammal-centric kind of approach to neuroscience that we kind of always have to obey, neurogenesis was thought to be more or less complete by the end of embryogenesis, that cells weren't going to be born beyond that. However, now, not only in mammals, but all sorts of other animals, and even humans, mammals, we actually still see neurogenesis well into post-embryonic development. So these are actual schematics traces of sections that were derived from human brains from hippocampi, from a brain bank. The blue just is an anatomical marker for the researchers, so you can see the elaboration of the hippocampus over time. And each one of those yellow dots is a recorded cell that's positive for two markers that together strongly suggest that this is a new neuron. Now, put that on a graph so it's a little more friendly to look at. It looks something like this. So even with human data, I will say human data, every year on PubMed, you can probably search and find a very famous paper that says adult neurogenesis is a thing in humans, and then like an equally popular paper that says it is not. It's an eternal fight. Well, over the years, what everyone has come to agree on is that postnatally and post-embryonically, there still is neurogenesis, even in humans, and even unique populations of neurons can be born through this. And so neurogenesis is something that certainly happens, and it certainly happens in the time window that we define as post-embryonic development. This leads to kind of a focus in my research, although since I've been here, I don't know, it gets a little more expansive. But in my postdoc, I certainly wanted to ask, does that mean neurogenesis itself actually mediates sensory experience dependent changes in brain development? So going back to our post-embryonic development or how we can kind of organize it, the animal I ended up studying this with is the zebrafish. And specifically, post-embryonic development for us really focuses on the larva. The age of the larva is actually unique to depending on the actual modality we study. So I did some work on vision, and our visual work actually doesn't start until we have a 5-day-old larva. The youngest larva is 3 days. That's kind of the transition to this period of life. And that was because at 5 days is when the eye actually makes contact with the brain. And that was the circuit we were interested in looking at. What I'm going to be talking about mostly today starts at 3 days, and for a couple of reasons that we'll get into. But it actually, so we really focus on the start of post-embryonic development. Realistically, this continues until 3 months when you get an adult fish, but there is a juvenile period at 1 month to 3 months. But the AUPs on that, whew. I worked on birds. I'm ready for a week-long experiment. So going back, so I've talked about these 2 critical traits that really define post-embryonic development. Do we see those in larvae? Is a good question. And so this is a 5-day-old live zebrafish larval brain. It is a transgenic line in which every brain cell expresses a calcium indicator, which, long story short, when you get a big influx of calcium, which is usually maps to activity in a brain cell, you get a fluorescent flash in a cell. This is from a lab in Genelia, Misha Aren's lab. And what you'll see is there's kind of this background level of activity, but ultimately this is an immobilized larva that will be presented with a visual stimuli, a very bright one in this case. You'll be able to tell, trust me, right there. And so this is a 5-day-old larva, there's a visual input. So in this period of post-embryonic development, we get sensory processing. The question is, do we get the kind of neuroplasticity I was talking about? Well, this is a picture from Ben Lindsay, who was in my old postdoc lab and is now a PI at the University of Manitoba. He came and gave a MCS talk for us. And he exposed fish to EDU. EDU is a synthetic version of thymidine. It will get taken up by any cell that's duplicating its DNA or replicating its DNA and thus undergoing proliferation. So we gave it enough time to just get taken up, sacrifice and process the brain. And what you can see is all these pink labeled cells were new since that EDU. And what you can tell is there's a tremendous overlap between not just sensory processing, but also the addition of new cells. So the 2 things we cared most about post-embryonic development. And this was the kind of evidence or orientation I approached my postdoc with. And I related 2 lineages of research in my postdoc. The first was just looking at the importance of visual input on developmental midbrain development, which is typically where the retina projects directly to, and also grows with neurogenesis. But what I'm really going to focus on today, and is really the study that carries me forward and motivates my research, is looking at motor experience, or experience just moving, or swimming in this case, and neurogenesis specifically in the forebrain. And we'll talk more about that right now. So, like I said, I'm sorry, lectures are done, but at least you get to be on this side of one. So this is a zebrafish larva in the post-embryonic stages that we study. If we took a coronal section, so we took a section through the brain and then turned it to face the camera, we can depict it like that. Now, I know, it's very ideal. This is a counterstain picture to show I'm not too exaggerating about what's going on here. So this is just a picture that's counterstained. This is just hook stained, so it labels all nuclei. Now you can actually divide the zebrafish forebrain into a dorsal pallium and an appropriately named ventral subpallium. They actually are provided and contain different types of niches that produce new neurons. And we're going to focus on my results this time on the dorsal pallium. We do have subpallial results, but I only have an hour. So how do new neurons contribute to the growth of the dorsal pallium? So ultimately, all new neurons have to come from some kind of neural stem cell, right? Some kind of originator. And in the dorsal pallium, it's uniquely this population of radioglia. They're depicted here in green, and then this is an actual picture of label it's a transgenic label, but then we labeled the GFP to kind of up it when we looked at sections of the brain. And so you can see they've got these pyramidal cell bodies that go along the top of the brain and down the midline. And then they have these huge radial processes that cut across, and that's why they're called radioglia. When radioglia divide, they do so asymmetrically. So they produce 2 different daughter cells. One of them is depicted down at the bottom is another radioglia. And then through this asymmetric replication, they maintain their own population. The other cell they generate is called a progenitor cell, intermediate progenitor cell typically. And it's labeled here on the right with an antibody for TBR2. It's a nightmare antibody. I think I'm one of 3 people in the world who got it to work. And then I stopped getting it to work afterwards. But what you can actually notice with the TBR2 label is there is a 1 cell level space from the labeling, the positive labeling, to the edge of the brain. And that's consistent because that's where the radioglia and neural stem cells are. Now, in the simplest situation, a progenitor simply differentiates. It turns into a mature neuron. We have mature neurons labeled in this bottom right with ELAB-L3. This is just a label, labels all mature neurons. And what you'll notice is there's an absence of labeling in the midline and across the top. And that's because that's where progenitors and radioglia are, which are not neurons. So it's a nice proof to us. Our current understanding of paleoneurogenesis in zebrafish actually suggests that a progenitor can do more than just passively differentiate. It can actually divide symmetrically. So one progenitor can produce 2, they might go on to then both differentiate. And through this kind of amplification via progenitors, you can get upwards of at least twice as many neurons born out of the same number of original divisions in the radioglia. And this is the simplified kind of version of dorsal paleo neurogenic brain growth. So the brain actually grows largely because of neurogenesis, with the idea that consecutive layers of new neurons are born on top of older. So you get an appositional form of growth, kind of like tree rings, if you want to think about it. And it stacks on top of each other. And this is how a large portion of the dorsal forebrain goes. Of course, there's other, migration of cells from elsewhere that come in, but this was the actual process we wanted to focus on. Maybe because we saw it affected by movement, I don't know. So also for the sake of simplicity, I'm only really going to focus on results from our study that used pCNA, which is proliferating cell nuclear antigen, which is just a marker of cell proliferation. It's not unique to neurogenesis, but we're looking at a neurogenic niche, and we did some other labels, so we can be confident that we are looking at neurogenesis. But because pCNA just labels cell proliferation, I wanted to go over what it actually is labeling based on this model. So it's actually labeling a combination of radioglia and progenitors, and specifically only the ones that are dividing. So what you can see in the schematic is that some pCNA, some radioglia will not exhibit pCNA because it's simply not dividing at that time. Either it's just not dividing at that moment, or it's quiescing. Which just not dividing is what that means. And it'll label progenitors, but only the progenitors that are potentially amplifying the signal and producing more progenitors. The ones that are just going to be passively maturing into neurons wouldn't express pCNA. And this is what a section of the brain looks like labeled for pCNA, so a simple readout. And as expected, you can see basically a 1 to 2 cell layer along the top of the brain and along the midline. And so everything I'm going to present, I think, until maybe later, is pCNA. So just for the simplicity of just giving us a metric of cell proliferation within the brain. So now we're finally poised, lecture's over. Now we can ask, can motor experience or can movement itself modulate forebrain neurogenesis? So we started this by, we had to manipulate movement in zebrafish, and we wanted to do so non-invasively, or minimally invasively, I guess, maybe, depends on your definition of invasive. So we took 3-day-old zebrafish larvae, and I'll really get to that later, but for this purpose, 3 days is where you can start seeing a zebrafish swim. It's very infrequent, however, it will get increasingly frequent day to day to day beyond this. So we needed swimming to be a thing because we actually need our fish to swim. We can't, we're not making those swim yet. We are here, but don't worry about it. So we raised our controls, just 1 apiece in 6-well plates, just the typical cell culture 6-well plates, no cells in it, obviously just water. And then I came up with this complicated contraction, which is I just put very small mesh fences around my restraint fish. The mesh enables them to access the same volume of water. However, only a very small portion of it is actually available to swim in. We're hoping the idea, provide less space, they'll swim less. And we sampled swimming to make sure they actually were changing their swimming. And then also, ultimately, like I said, I'll show you guys the forebrain pCNA data at both 6 days and 9 days. So a 3-day treatment and a 6-day treatment. So this is our swim data. We use automatic tracking. On the left, we have an example of a control at 4, 6, and 8 days, the same control. And we just have a track. This is around an hour of swimming, I believe, and our restraints. So if nothing else, you can see the restraints were restrained. However, it's not particularly easy to read into these things. So we have it all graphed on the right. So on the x-axis, we just have the age, 4, 6, 8. And on the y-axis, we have corrected for the number of hours we recorded them. We recorded them for 8 hours, I believe. What was the average hourly distance they swam? So at day 4, we didn't see any difference. The red is the restraint. The black circles are control. However, from 6 days onward, we saw a significant difference where restraint kept those fish swimming less, while the controls could swim more and more. So we had successfully done it. And so now the question is, what's actually happened with cell proliferation in the dorsal pallium? So this is at day 6. So on the x-axis, control and restraint, again, black and red relatively. On the y-axis, we have the number of pCNA cells. We sampled across 3 to 5 sections. I think for this one, we did 3 sections, centered on the dorsal forebrain. And this one, we actually also take the pCNA cells. We can use anatomical markers to isolate the dorsal pallium. And we corrected for the number of total cells there, just using our hooks or our blue label of nuclei. And what we saw in the graph, but you can also see with the representative images, is already after 3 days of restraint, control fish exhibited significantly more cell proliferation in the dorsal pallium compared to the restraint group. Perhaps it's not too surprising then, if we continued the experiment till day 9, we saw an even kind of more exaggerated effect. So same kind of graph, the restraints are still lower, they're even kind of crushed together even a bit more. And you can actually really see in some of these brains, it was really stopping, at least the expression of pCNA. Whereas it was still going strong in controls. Something else you'll notice that I don't think I show the data on, by day 9, we're actually getting different sized brains. So we're getting smaller brains in the restraint versus the control, which we didn't get at day 6. It seems like a longer term effect of development under restraint. Now this was great. We were excited. We found, you get excited anytime you can put a picture instead of a graph. That's awesome. However, there's a huge amount of limitations to this paradigm, right? We put a little fence around the larva. It doesn't move, but it also doesn't do potentially a lot of things. It doesn't see as much of the world. It doesn't actually get to physically experience as much of the world. So we sought to complement this approach with an opposite. Could we make the animals swim more and ask if that can upregulate pCNA? And here comes more of my engineering, if you want to call it that. I cut a tube in half. And then we put it on the water system with our zebrafish. So we had 2 groups. We had our control group. So these were group reared in these swim tunnels. That's a very generous terminology. But in the controls, basically throughout the day, we adjusted water flow. There was always water flow, but it was never fast enough to actually physically displace any of these larvae. So they never had to swim to maintain their position. Whereas in the current condition, the exact same tunnel was used, but we modulated throughout 2 periods of swimming per day, we modulated the flow to make sure that it would be the least amount that would start displacing larvae. And I'm sure there's a lot nicer thing that you could build with that, but I'm not the person. So we repeated this and we went to day 6 and day 9, again, starting at day 3. Okay, so at day 6, we have the data for both here. So day 6 is in the left graph, day 9 is in the right. And what we saw was in both, when we looked at pCNA, again, corrected for the number of cells. I will say, if anyone had a question, we did both corrected and uncorrected analysis for all of our work, and none of it ever disagreed. It was always showed the same whenever we saw a difference. And what we saw was that by day 6, and then also by day 9, we saw a smaller but a significant increase in cell proliferation in the same region where we saw it reduced under restriction. So these experiments together made us feel pretty confident that we could conclude that motor experience, or the physical experience of movement, actually modulates post-embryonic development or neurogenesis in the dorsal pallium. It does so negatively, again, reduced swimming, reduced forebrain cell proliferation, and positively, increased swimming and led to an increase in cell proliferation in the forebrain. We did some other labels, and I'm more than happy to talk about them, but I haven't put them in the talk, where we actually believe what's happening is a modulation of that progenitor population, the proliferating cells, and the number of new neurons they actually bring. And specifically what we think we would see is that proliferation was actually comparable at the start by day 6, but more of the cells that were being produced in the restraint were just turning into neurons. Whereas in the control group, they were actually producing more progenitors, before they differentiated into neurons, we think. Now, we wanted to do so I was new to zebrafish, I was new to this model, and my supervisor says, oh, well, you want to publish this, you have to figure out what the mechanism is. and I was not a geneticist, and I didn't understand what that meant. And so my approach to the mechanism was to ask, well, what is movement? Movement is a multisensory, very complicated behavior. What does that mean as sensory feedback to the animal, or any kind of feedback to the animal? Now, we were really inspired, or I was at least, by the work from the 90s onward, where they get rats, adult rats, to run in a wheel, and then it increases neurogenesis in the hippocampus. That's a legacy work. I always loved it. And at the time, the big thing that people were looking at was circulating factors that were released from the muscles. So things like irisin or myokines that they get into the blood when the muscles are being used through exercise and somehow get up to the hippocampus and modulate and upregulate neurogenesis. However, this early on, I kind of viewed what we were doing as movement as much more rudimentary and fundamental to life, not an enrichment with exercise. So I wanted to know, does sensory input, a direct, piece of input picked up by the nervous system, stay in the nervous system and actually modulate this change in neurogenesis? So I did kind of these slides so that I hopefully could talk a little bit about what we've actually done here. But I will go over, so me and my boss, Vince, we sat in his office, we pretended to be larvae, we imagined swimming through, I won't lie to you guys, the Petri dishes we used slightly less flora than depicted here. But one thing when you imagine you're swimming through is visual input's a big thing. So optic flow is the name we give the phenomenon when the visual environment moves past us while we walk through it, for example, or swim through it. Sorry to break it to you, we didn't find any evidence. Like I said, I could talk about it if you want, but I cut the data. We didn't have evidence that visual input was responsible for any of this effect. But we did find in the same experiment that physical movement physical movement itself alone, without any visual change, could repeat our effect on increasing neurogenesis. So we knew we had to think about a sensory system that made sense physically. Now, this one's a little specific to amphibians and fish, but zebrafish have a sensory system that can detect water flow called the lateral line. This is a transgenic, which shows different components of the lateral line. And if you zoom in on one of them, you see it's actually this little bundle of hair cells that just stick out into the water. Now, they're very much like the hair cells that you have in your vestibular system or your inner ear, in that when they bend, they fire. They cause firing in the sensory cells, or firing in the afferent fibers that innervate them at least. So we did an experiment. Because they're so exposed, you can actually ablate them very easily. They will regenerate, but we ablated them for a couple of days, and we saw it had no effect on neurogenic rates in the forebrain. So rule of threes, of course, the third one we would go to. We looked within the animal for a somatosensory form of input. And so zebrafish larvae have at least two forms within the body, or innervating the skin of the body, that produce somatosensory input. So the sense of touch, but also these are also neurons that are involved in chemosensation, so chemical sensation, pain. I don't know if you could say zebrafish have something like proprioception, which is the idea you can close your eye and know where your arm is and bent. But we think they might, based on kind of how they work in our experiments. So this is just a transgenic line, which labels, among other things, the two types of somatosensory systems in zebrafish. I had this in Toronto, and thanks to Ted, I also have it here, stolen from his lab. So there's two types of populations that process somatosensory input in zebrafish larvae at this age, which is 3 days onward. In the spine, there's a group called Rohan beard cells, named after at least one, maybe two people. I'm not sure, admittedly. And they innervate the skin. They detect all sorts of somatosensory input. But what's unique about them, we don't have Rohan beard cells, and they actually develop within 1 day. So when you come to your embryo the second day after it's been one cell, it will already have Rohan beard cells. They actually peak in number at 2 days, and then throughout the rest of life will slowly die off. There's a really interesting transient somatosensory population that really functions most strongly during the embryonic period and then becomes kind of obsolete. Now below them, we have dorsal root ganglia. Now these don't develop until day 3. So this was also kind of a suggestion to us that this is actually maybe a meaningful pathway, because our experiments in the swimming itself really mirrors the development of dorsal root ganglia. Now we have, all vertebrates that have ever been studied have dorsal root ganglia. This is just kind of a collection of cells that innervate the skin and detect all of those things from the body. Dorsal roots first appear at 3 days, and they'll continue to grow, including the addition of new dorsal root ganglia, all the way up to adulthood in zebrafish. They're usually done much more early in mammals. So we have two populations. are there any kind of manipulations in the literature that were available that we could actually modify them to test if they're involved? And so we can actually block the development of dorsal root ganglia. This is some work pioneered by Judith Eisen, who's a huge inspiration to me in this field. So we have a beautifully named drug, AG-1478. I'm sure you guys know worse, I know, but this is the worst one I got, which is an antagonist for the ERBB receptor, and ultimately can be used strategically to actually block the development of dorsal roots altogether. So it's a specific 8 to 30 hour post-fertilization treatment, and then a washout and never treat the animal again. So what's happening during 8 to 30 hours, as I mentioned, the dorsal roots don't actually appear until day 3. The precursors that will ultimately reside in dorsal root ganglia and in dorsal roots and produce those ganglia actually migrate from the spine, following those dotted arrows I put on the bottom. Now, ultimately, they stop somewhere and then know to just kind of set up shop and start producing cells. And part of how they stop is they actually use ERBB signaling. So if you interfere and block ERBB signaling, they continue to migrate, and they migrate apparently and end up at the bottom of the body. And when they stop, they don't have those local signals they need to know how to actually differentiate and start producing dorsal root ganglia. So they produce what I think is really incredible, which is if we have a normal DMSO vehicle, you have both Rohan beard cells and dorsal roots, but we can produce larvae that have all the Rohan beard cells, but lack any dorsal roots along the body. So it's incredible manipulation for us. Judith Eisen's work's incredible on this stuff. So the experiment we did is now we have 8-hour embryos, and we treat half with AG-1478 or DMSO. Again, very low concentration, very brief period of time, and then never again. And with each group, we repeated our control and restraint experiments, since we could produce pretty reliable, robust effects. And then collect and process brains at 9 days old. Now, we had to do the scary experiment first, which was, we're getting rid of a sensory system. So is this going to actually affect how much our fish swim? Because we're really relying on them swimming when we want to ask, is swimming important? So this is some swim data from day 8. So again, on the y-axis, we have our distance swam. I always use the wrong word. And on the bottom, we have our 4 different groups. So the first 2 groups are just our controls, so they all have dorsal roots. And then we have our control and restraint, and we can see a replication of what we found before. Restraining fish reduces proliferation in the forebrain, and reduces swimming. And then second, we have our fish in red that lack dorsal root ganglia. And what we show is not only does restraint reduce the amount of swimming, but more importantly, the amount of swimming between our control groups is comparable. And this is consistent. Judith Eisen, she didn't actually quantify it, but she actually raised fish like this to adulthood and reported that she's never noticed any kind of like strange behavior or swimming or anything. There's some cool stuff we want to do with that too. So when we look at PCNA at day 9, this is what we found. So just walking through it, the first 2, again, is a full replication of what we found before. Restraint in the presence of dorsal root ganglia decreases PCNA. Interestingly, when we look at our fish that lack dorsal root ganglia in red, we see that restraint still actually significantly reduces proliferation in the brain. But the big one here is comparing those 2 control groups. In the absence of dorsal root ganglia, so in larvae that don't develop them, the same amount of swimming no longer translates to the same amount of cell proliferation in the forebrain. And these are just 2 representatives between those groups. So we had our first evidence that we actually identified at least one pathway, not the only one, because again, restraint is still decreasing proliferation in the brain, even when dorsal root ganglia don't exist in these animals. So with dorsal roots in part, we concluded mediate or appear to mediate movement-dependent forebrain growth. So larvae lack them, exhibit reduced movement-dependent neurogenesis. Again, same amount of movement, but less neurogenesis. Now we reasoned, again, to complement this approach, we reasoned that if dorsal root ganglia are important and their activity mediates this effect, if we could activate them in the absence of movement, so in completely immobile larva, could we metaphorically trick the forebrain into thinking it's getting a bunch of sensory input and produce cells, even when the animal is actually immobilized? And for this one, we leaned on another incredible paper by David Kokel, which is the identification of a small molecule called optivin, pictured here on the left. Optivin binds to a surface sensory receptor called TRPA1. I work with the Helios, so of course we have 2 versions of that. And in zebrafish, it binds specifically TRPA1B. TRPA1B is expressed in dorsal root ganglia, in Rohan beard cells, and in trigeminal cells. So everything that's somatosensory. And it's actually responsible for transducing a lot of different, like chemosensation, pain, and even hardcore mechanosensation. But what's incredible about Optivin is it will bind to this surface receptor, which also acts as an ion channel, so it's ionotropic. And if Optivin's around bound to TRPA1, and you actually expose it to light, expose the fish to light, specifically violet light, but broad light, white light has enough violet in it, it will open the channels. So it's kind of like an optogenetic, but without the genetic. It's actually a drug that acts like a light activated channel. And so just to show you what maybe a little extreme version of that looks like from David Koppel's work, these are some larvae. They're still in their chorions. We usually hatch them, but the drug gets in no problem. They're soaked in it. And what you'll see is a flash of light to activate it. And so you see a huge behavioral response because you can imagine you're activating every somatosensory cell. So it's going to trigger a huge behavioral response. This was a little extreme for us. This is not what we wanted to do. So instead, this is all recorded in infrared in case you're wondering why you can see it without it being activated. So we actually came up with a paradigm where we could kind of more safely, transiently, and acutely activate Optivin repeatedly over longer periods of time without compromising the health of our larvae. So this is a behavioral recording from a plate of Optivin-soaked larvae, which is the blue, and DMSO vehicle-soaked fish in gray. Now, what this paradigm we ultimately set on is every 5 minutes, there's 2 seconds of light that would activate Optivin, then 5 minutes of darkness. And so just an alternation between that. And we found that we could do this for at least 5 hours. We didn't go beyond that. And the fish would be completely fine when we rinsed them out, freed them, put them in a bigger arena or anything like that. And just to show, this is what an example does. So if you can see in blue, these yellow arrows are one of those 2-second flashes of light. Each point is a 5-second bin of behavior. So that's why they don't swim too much per dot. But this is an example trace of an Optivin-soaked fish before, during, and after 5 second, 5 second, 5 second, one of these light pulses. Whereas if you look in the background at the gray, you can see those larvae do not care about the flickering of light. So, like I said, we had to immobilize the animals. So for this experiment, we took 3-day-old larvae and immobilized them in agarose in 24-well plates, complete immobilization. And then at day 5, we kept them just in the dark, I think, normal development. And at day 5, we did a 2x2 experiment where each 2 trays got Optivin, 2 trays got a vehicle, and then 2x2, whether or not they were exposed to dark for 5 hours or our light pulse schedule for 5 hours. Then we rinsed everyone out in the dark, let some time for Optivin to wash out. Then we put everyone away for 12 hours. So we are asking just within 12 hours, are we seeing any effect on proliferation? So this is the results. So again, PCNA in the pallium corrected for hooks. These are just our DMSO groups. So no Optivin present. And we can show our light schedule by itself, sadly, did not do anything to proliferation in the brain. Alternatively, with our Optivin groups, the combination of Optivin and light showed out just 12 hours later after the 5-hour treatment, already a significant increase in cell proliferation in the pallium. Now, this was very exciting. However, again, I kind of mentioned it before, but TRPA1B, the actual receptor itself, isn't just in dorsal roots. It's in Rohan beard cells, it's in trigeminal cells, which those innervate more or less the head and the neck. And so what we had to do was combine literally everything we knew and repeat this experiment, but only use the active conditions, so only use light and Optivin, but now repeat it in our fish that no longer have dorsal roots versus the controls that do. So this is just the graph I showed you before. So what do our results look like when we just repeat it? Everyone gets Optivin, everyone gets light tracks, but some have dorsal roots and others don't. So our control group showed the exact same increase in proliferation in black here. And the red group that lacks those dorsal roots returned right back to a comparable level, despite receiving both Optivin and light, as all of our other groups in our previous experiment. So in conclusion, movement modulates forebrain growth during post-embryonic development. Like I said, I didn't show the data, but we think it's because by regulating that progenitor population of cells. Somatosensory, but not visual or lateral line feedback via the dorsal roots, at least in part, mediates that movement dependent for brain neurogenesis. And at least at the time, but I think something that kind of undergirds our interest in this whole approach is that it might actually represent kind of a unique form of sensory coupling and via sensory feedback between the body and the brain. If you think about the body and movement, and that's a developmental process, right? You're not born walking. And at the same time, to know it's actually providing more growth to the brain, perhaps that can feed back and actually help control future behavior. And we were particularly interested in it because with statistics for people who are experiencing mental health disability, one of the most common comorbidities or co-occurrences is physical disability. And even here, you can actually see there's also developmental disability. And so we're wondering if we're actually looking at something that's so basic that it might actually explain an interaction between development of the nervous system and the body, or at least the musculature. Okay, how are we doing? I didn't have a timer. Oh, okay, we're good, I think. So, as a very, very, very light taste, I don't want to spoil my my students' results, and they have very cool stuff. Some of it I do want replicated, don't worry. And so I thought I would just talk about what my three kind of grad students right now are kicking things off with. So one of my master's students, Shelby, is trying to actually ask maybe a pretty important question, which is how does that motor experience get to the brain and impact neurogenesis there? Now, I'm actually going to show data that has nothing to do with that, but because she wants to ask these questions more molecular, more genetic, more cellular, she wants a paradigm that's faster. You know, I mean, you might have noticed when I showed you how much the fish swim, if you give a fish a control well, some of them will swim, some of them won't. There's a huge amount of individual variation. So Shelby's trying to develop some kind of protocols and paradigms where we more reliably can get a stauncher kind of no overlap difference between the two. So there's Shelby. And so her first one is immobilization with methylcellulose. So agarose, like I used, it's a nightmare. It's not fun. I don't like doing that to animals. Methylcellulose is much more forgiving. It's basically you dissolve it and it's a very thick water. I know it's a weird thing to say. And people use it for microscopy, for example. But we found that if we bump up the concentration to around 6%, or I should say Shelby found out, that this is just a swimming graph. So this is just total swimming during a recording at days 3, 4, 5, and 6. So you can really see the blue is the controls. Like I mentioned, 3 days is really the kickoff of swimming. And it only gets more common day to day to day. And 6 typically is the peak we see with the trays we use. However, believe it or not, there's 24 controls here. There's 24 embedded fish down there. I mean, it's probably not too surprising, but when we mobilize them, they all basically don't move whatsoever. So a true immobilization. But they look great. They survive. You can free them later. They'll swim totally normally. So it's a lot nicer than something like agarose, which is very stiff. And the other one, again, thanks to Ted for putting me on this track, it's an actual CRISPR approach where we inject the CRISPR kind of materials, and it actually is so efficient, we can study the fish we actually inject. We phenotype them to make sure though, which is we use a previously published CRISPR guide that targets CHRNA1, which is part of the nicotinic receptor. So the nicotinic receptor receives the neurotransmitters from motor neurons. So it's the way muscle responds to motor neuron pulses. So if you interfere with it with this published CHRNA1 guide, the fish are always immobile. They're incapable of responding to neural impulses with muscle contraction. And if you weren't sure, that there's some proof for you. This one's only 12, but all 12 are in that red line. And so the advantage here is that Shelby's produced 2 very different but complementary approaches to immobilization. For example, if we want to add something or if we want to fish in completely normal water, we can use our mutant instead of having to deal with methylcellulose, which might interfere with the addition of a drug, for example. And then the other advantage is that we can use normal non-mutant fish when we use methylcellulose and not have to deal with a mutant who's been a mutant its whole life. And so that's what Shelby's doing. Shelby's also, actually everyone in my lab, something else we're getting away from that I've shown a lot here is we're stopping sectioning the brain whenever possible. It very much limits where you're looking in the brain. We looked at the forebrain for some like social reasons almost. Like that's where everyone would expect neurogenesis to be affected because of the mammalian work. But in reality, we want to know what the whole brain is doing, or as much of it we can look at. And so we're also taking an approach where we're actually dissecting out larval brains, which was a couple months of nightmare, but they're getting it. in part, I had to buy better forceps, so I'll take that one. And so on the left here, we just have a counter stain. This is a complete zebrafish larval brain. And on the right, on the red channel, we have an antibody for phosphohistone H3, which is a very acute marker of proliferation. So there's a lot fewer cells than with PCNA, but we're also planning to use PCNA with this approach. But as you can probably tell, PH3 is actually very conducive to counting, which is very nice. So second, my PhD student, Gabriel, he's actually studying if somatosensory feedback impacts the development of the peripheral sensory nervous system. So that's Gabriel. He's using my restraint paradigm, which is kind of depicted on the right. Something we've noticed very interestingly is that here, and I don't know, it might be the fish, might be our trays, something, we still see that peak. And so it's a swimming chart. And so at day 6, again, we see this huge peak in swimming. But at day 9, a lot of our controls actually swim less. I don't know if it's a habituation, or maybe I actually have a sneaking suspicion that when you're getting to day 9, our control wells become restraint wells. You know what I mean? This is probably impeding the animal's development beyond that. But we can't fit that many in the incubator, so I don't know. So we have some ideas. And what Gabriel wanted to actually point out was that, you know, I focused on this forebrain, that ignores that there's neurogenesis all over the place in zebrafish in the nervous system. But most importantly is those dorsal root ganglia I told you about themselves undergo neurogenic development. They add new cells. And so based on some preliminary data I got from my postdoc, Gabriel's taking the lead to ask whether or not reduced swimming actually, this is a very circular question, but does restraining or reducing swimming impact the development of the cells that would then detect swimming? It's a little circular, but I think it's a really interesting concept about how the animal is actually interacting with its environment and how development itself fits into that. And this approach is, again, looking at the tail. So this is that IL-2B GFP line again. But we're producing some other ones, not only so that Gabriel can actually count them in process tissue, but ideally so that Gabriel can actually count the same fish multiple times non-invasively using fluorescent markers. And finally, the question is brain development affected in diseases that are characterized by compromised motor development? So everything I've shown you is quote unquote a normal fish that we've made swim differently. But we recognize pretty soon that a naturalistic example of a fish that's not going to swim normally early in life is one that suffers from muscular disease. So my master's student, Micaiah, but realistically, a lot of this work done by a really incredible URI undergrad and 399 student, Peria, who's sadly leaving my lab. Apparently like medical school is important or something. And also, sorry, Micaiah is camera shy. So they've actually looked at our first disease in the lab, which is mutations in the gene nebulin, which is a big structural gene in muscle. And diseases associated with it are called nemeline myopathy. So we've done stuff like look at the muscle, genotype, to have evidence that we're producing mutants pretty reliably. This is the swim data. So in blue, we have our control injected. We have a CRISPR injection control. And in red are our fish that are mutants. And so you can see a significant reduction in the amount of swimming. And here you go. This is the number of PH3 or phosphohistone H3 positive cells per forebrain. And in our preliminary data so far in Peria's thesis, we saw a significant reduction in the amount of cell proliferation occurring in the forebrain of mutants compared to controls. And that's just a little touch. Well, like I said, we're all starting, and this summer is our first summer together, so I'm sure it will be a little crazy. And with that, I just wanted to acknowledge everyone that's helped me. So my whole lab, Gabriel, Arash, Shelby, Micaiah, Peria, Kayla, my U of T collaborators, my old supervisor, Vince, James, who I started a lot of the he studies muscular development in zebrafish, Ashley, Bruce, Todd, the U of T staff that helped, and then the providers of a lot of the transgenic lines I actually showed today. And with that, I can take any questions that anyone has.
Great, fantastic. Thanks so much, Zach. So open for questions, but we should see if there's any questions online here. So I can just escape this.
I don't know where I put it. They give you go up to the bar at the top. And then you can hit chat. Chat square. They're all sleeping.
Okay. Okay, so
Yeah, really, really cool work. Thanks for your talk. So during your critical windows that you're looking at, so I guess days 3 to 9, during that time, like, have the dorsal root ganglions already developed their dendrites and axons, and have they reached their like tissues, like what, like where they?
Yeah, absolutely. It's a great question. Yeah, they do very quickly because overwhelmingly they innervate the skin. And so they're literally born microns away from where they'll actually innervate. They have a very unique innervation pattern. So when I showed like the tail and you saw like a bazillion little axon terminals, those are mostly Rohan beard cells, which have this kind of cloud innervation. But dorsal roots themselves actually have a pretty stereotypical one. And it starts almost immediately. Like once we can see them with our fluorescent tool, our fluorescent marker, you can actually see the axon terminals as well. But we can also show that they're active when you actually activate or move the body.
So their axons are in the skin.
Yeah.
And then their dendrites are where?
Oh wait, sorry. Geez, I got it backwards, of course. Sorry, yes. Yeah. They innervate, the dendrites innervate the skin, and then the axons go into the spine. I actually have some very preliminary work because a huge question for us is, how is this information getting to the brain? And as far as I can tell, we do have plans once we have a confocal that we can do in vivo imaging with to actually try to produce a kind of connectome. But so far I've done like mosaics to isolate cells, and overwhelmingly they all terminate in the hindbrain. So they go directly into the brain, but they stop at the hindbrain. So there's super interesting questions about how that actually would get to something like the forebrain as well.
So that was a great talk. I've learned a lot about techniques for immobilizing and restraining fish, so that was great. But I got the impression that the fish weren't swimming in a very visually rich environment. Like if you've got a fish in a swim tunnel in particular, it's just swimming to maintain its position. It's not seeing much as it swims around. So if you had the fish swimming in an environment where there was more to see, do you think you might even get even more neurogenesis?
I suspect with the history of environmental enrichment and neurogenesis in all animals studied, absolutely, the more you throw at them, the better. Visual input takes a very roundabout way to get to the forebrain, but absolutely, if you focus on the midbrain, for example, and we've even started looking at the midbrain because we have the whole brain, I absolutely think that. We've even talked about, this is kind of a pie in the sky because I have no idea what would happen, but we've even talked about making like different configurations of the same area of swimming. But I actually think that might be a non-invasive way to make them swim less, because I have a sneaking suspicion with certain kind of arrays of orientation, they wouldn't swim as much. But I couldn't tell you why behaviorally. I wish I could, but zebrafish larval cognition will be on me. I left that behind in birds.
Well, yeah, a question about whether you think the fish are stressed when you immobilize them. And related, this may be related to that, do you have a genetic CRISPR line that swims more? So I mean, I think the gain of function is really important there, but do you have that in the genetic world as well?
Yeah, I'm not like I'm not so much of a, what do I want to say, a genetic like determinist. And so when we want to make our fish swim more, we wanted to do it in some kind of like non-invasive way that we could use for multiple models. We're reinventing the swim tunnels, and I'm hoping a little bit more friendly of a way with peristaltic pumps, with the idea being it might not even be consistent flow, but they actually don't swim consistently. They do something called beat and glide, where they beat their tail and glide. And so, what was your first question? Sorry.
Oh, when you put them in.
Oh, stress.
Or when you think that.
Yeah, absolutely. I would suspect that maybe. I think with something like the restraint, the advantage there is that the fish, as long as we've seen them, develop normally, they look great. I think something like mobilization is a little tougher to resolve that. And yeah, I don't know, it's tough. Like, and this is exactly what I was talking about Corwin about, is like, I actually wonder if there's something perceptual about the arrangement of an environment where we could actually give two fish technically the same amount of space, but they wouldn't use it the same way. But who's to say that stress could also be mediating it? Yeah, absolutely. No, it's a great question. And it's like one of those things where our solution tends to be to come up with some weird experiment that hopefully does the opposite. But, or even, you know, sampling cortisol, like if you're okay with that shorthand, I don't know, I totally believe that a measure of like corticosterone is a one-to-one representative of stress, but that would be presumably the physiological mediator most people would suggest. Yeah, it's a really great question.
If I can sneak one in before John. So just to follow up from Anna's question, so the DRG cells, they aren't just all homogeneous, there are different DRG cells in there as
Absolutely.
well, right? So do you have any idea what you might like, if any specific sets of cells might be involved, than more so than other ones, or how you might target that?
It's tough. It's tough because, you know, approaching this is we can only isolate them insofar as we have markers to isolate them, and every antibody we could use will label them all. Albert Sagasti's lab actually produced a bunch of new enhancers and promoters, but they all just again label dorsal root ganglia or not. We could produce mosaics, but in my experience, at least on the cell inside the body, if you're focusing on the body, they look very, very similar, even when they're different parts of the same ganglia or same root. But I do suspect that they probably terminate elsewhere in the brain. So I guess I can just say the whole thing. The hope is that we're making a line right now that has a photoconvertible protein that's expressed in all these cells. And the idea is if we can get a microscope capable of it, that we can individually photoconvert, so switch an entire cell from red to green, then give it the time for that photoconverted protein to get through. And that way we would actually be able to compare different cells within a root and ask, yeah, but right now we have to hit it with the sledgehammer of all or nothing. Yeah, absolutely.
Okay.
And it's even, to be honest, it's even within a cell, there's so many different functions served by the exact same cell even. Yeah.
John?
Yeah, a very general question. Now, so you can induce or enhance progenitor cell proliferation in general. But depending on, let's say, the specific input you do to enhance those increased generation or division of those progenitor cells, do they then subsequently target themselves into neuronal circuits that are preferentially designated for one thing or another thing?
Yeah, it's an incredible question. And it's like, so we have some extra data. We have data in the Eli paper just showing that they're all neurons. Not surprising, you know, not too surprising, but absolutely. So for example, speaking to your question, when Ben Lindsay did some work on sensory experience in adult fish, he actually showed that there was niche specific responses depending on the modality of the enrichment. So smell versus social enrichment and visual enrichment. And I absolutely would suspect that we're producing, if nothing else, potentially different types of neurons. Again, this is like the limit of, you know, what markers can we use? And so we can use a marker that shows it's a neuron, not particularly helpful. Zebrafish lack a really good antibody for glutamatergic neurons. Glutamatergic neurons are overwhelmingly produced in the dorsal pallium. However, I think I got a lead on the private antibody in someone's lab, but even that, I don't know if it would be different enough. The approach we would take is we would use EDU and at the start of our experiment and ask where those cells end up. Absolutely. It's an incredible answer. And I think by getting out of sections, we can actually look for those cells elsewhere in the brain than just in the immediate vicinity of where they're produced.
Kathy, and then we'll probably stop there.
So I'm just wondering about the variability that you see in your controls, and if you've ever gone and looked to see if that actually correlates to brain differences.
Yeah, it's something me and my supervisor, like from my postdoc onward, it's like a dream that we've always had. The nightmare is isolating a single larval brain and processing it through immunohistochemistry, isolated, you know, one by one. It would probably like, you know, increase the amount of reagents we need by like 100 times, but I'm still set on it. And if nothing else, I think we could probably make some kind of like mesh where you can isolate the brains and kind of dip them, the reagent to reagent. Absolutely. And I think the solution that we might come up with, hopefully that would be the best, is if we can have an actual neuronal marker, if of proliferation. There are some floating around, but I've never seen one, and I don't think they work as well as people had hoped. But if we can come up with some kind of non-invasive fluorescent way to actually sample anything then we could actually just image them all post-experiment non-invasively. And I think that would be the best way. Yeah, and we would be able to do that with dorsal roots. So if we see that difference, we would probably look at that. Yeah, great question though. It's, because, we want to embrace individual variation because it's crazy when you let those.
Makes me wonder if there's like a whole bunch of them that are inbred or.
Sure, that's probably the same. Well, I think it's really interesting because it also speaks to, it like bizarrely speaks to an animal's agency in its own development. Which is like, I don't know what you do. I'm too simple to understand what you do with that concept, but it's a really great question.
Fantastic. So with that, I'd like to say thank you very much for an excellent talk today, Zach, and for finishing off our Chairs lecture series. I haven't seen anything on the chat, but lots of questions here today. So thank you very much. Token of our appreciation, and I have a couple of things here. As the last talk, we also today just received some new swag. And so here's a lunch bag.
Oh.
This we've been doing with everybody, so this is a constant. So yeah, thanks.
No, no problem. Thank you so much.